Thanks so much. And I really appreciate the invitation to come and talk to you today. My group is at MIT, and we work on technologies for mapping and trying to repair the brain, which of course is incredibly complicated. And both because I'll talk about them, but also as my conflict of interest disclosure, I helped found two companies that are trying to take some of these ideas and help see if we can help people. Now, it's useful to take a step back and ask, why is the brain so darn complicated? And one issue is spatial scale. You know, brain cells, neurons, are by far the largest cells in the body, right? They can be centimeters in the brain, down to spinal cord, a meter in length. And yet, the building blocks of the brain, molecules, synaptic connections, proteins, and so forth, are incredibly, incredibly tiny and organized with nanoscale precision. So if you took a human brain and could somehow blow it up to be the size of a city block, the synapses or connections between neurons would be the size of grains of sand. And the molecules, the proteins that make those synapses communicate are incredibly small, nanometers in dimension. So how do you deal with these incredibly large spatial scales? And if you want to understand something like a feeling or a thought, you're going to have to take into account large-scale circuits. And the same is true for looking at diseases. How can you pinpoint the cause when there's so much possibility going on? To make things worse, there's time to deal with as well. So if you think about what the brain does and how it goes wrong, whether it's forming a memory or the progression of Alzheimer's disease or learning a language, these processes can take years, sometimes decades. But the briefest events in the brain, short electrical pulses that last a millisecond in duration, are incredibly high speed. And of course, you have to couple that to the incredible density of neural computation. So within a cubic millimeter of your brain, you're going to have about 100,000 of these neurons or brain cells, and they're going to be operating with these high-speed electrical pulses. So the vast differences in temporal scale and spatial scale that you have to deal with really put brain science into a very difficult category to even understand what's going on, much less fix what's going on. And of course, we want to not just understand the brain for philosophical reasons. Brain disorders remain largely uncured, and in fact, for many brain diseases, there aren't really even any really good treatments. And part of the problem is, of course, this complexity we're talking about. So if you build a drug to try to treat a brain disease, these are some numbers from 2010. I think it's a little bit even more pessimistic nowadays. But from an editorial in Science back 7 years ago, they reported that brain drugs take longer to develop than any other category. The failure rate is over 90% to get through clinical trials. They cost an enormous amount of money each. This number, I think, has also gone up. And even when they do make it through this pipeline, the efficacy tends to be partial, and side effects are nearly universal. So today I want to tell you two short stories about space and time, and how we're trying to build tools to help brain scientists cross space and time, and how we think that these can help improve things like diagnosis and treatment near the end. So let's start with space. If we want to understand how brain cells are organized in these complex circuits to generate things like feelings and thoughts and so forth, and how they go awry in brain disease states, we have to be able to image molecules within brain cells and brain cells in complex three-dimensional circuits. And that's really challenging. So microscopes, of course, have been around for a long time, hundreds of years, and they've been very powerful in neuroscience. In fact, this is how Ramonica Hall discovered the neuron in the first place and characterized the boundaries, the connections, and so forth. But the problem is the microscopes are limited. They're blurry. You can't see these finest connections and molecules, which are too small, much smaller than the wavelength of light that you use to see things in the microscope. So in our group, starting really about 5 years ago, we started thinking about responsive polymers. And what are those? Well, you have polymers that are chains of molecules that are all connected end to end. And if you take a polymer like the kind that you might find in a baby diaper, and you add water, what happens? Well, the water's drawn in through osmotic force, and all the polymer threads in the baby diaper material will spread out and swell. And these polymers are also highly charged, so they're going to repel and swell even bigger. And so this is an experiment that millions of kids do every day. But we started thinking, what if we did it on purpose inside brain cells or brain tissues? So the idea was, let's install chains of these polymers so they wind their way around biomolecules and between biomolecules. And if we do it just right, maybe we could push all the molecules in all the cells, in all these circuits, apart from each other, and move them apart until they're so far apart that you can actually tell them apart, even in a microscope that has physical limits that cause blur. And furthermore, if we make room around the molecules, maybe we can run interesting chemical reactions, like sequencing of nucleic acids, to figure out what makes those cells the way they are. So we call this expansion microscopy. And we've published a couple papers just in the last 2 years on this technology. And as I mentioned, we spun out a small company to try to get this into the world as well as in parallel to these publications. So if we do it right, maybe what we can get is take a brain cell like the one on the left in this cartoon and push the molecules apart so that, like stars in a constellation, they're going to be suspended in space, of course, anchored by this polymer. And the relative organization, though, would be the same. So if we were to look at this, it should look just like a regular cell, but with finer resolution. That's our goal. So to do this, we had to invent 3 new kinds of chemistries. And so let's pretend in this cartoon that we're zooming into individual molecules, these brownish blobs. The first thing we have to do is to attach anchors to the molecules so that we can apply force to them and pull them apart. And so we've developed anchors for DNA, for RNA, for proteins, and we're working on other biomolecules as well, so we can attach these covalently and then apply force to pull them, hopefully gracefully, apart from each other. The second step is, how do you form these swallowable polymers? You can't just dump baby diaper material on top of the brain. We tried. And if you put just as a joke though, if you can wash in the monomers, the building blocks, and have them form chains through a process called polymerization. And when the chains encounter these handles or anchors that we developed, then they connect. So now we have the 2 building blocks, the polymers that will swell, and the anchors that the biomolecules would ideally come along for the ride. And then thirdly, we had to find a way to loosen up the mechanical properties of these tissues. They don't like to be pulled apart, and we came with all sorts of tricks to do that. When you can add water, the polymers will swell again, just as in the baby diaper case, but now, through those anchors, the biomolecules will accompany the polymers. And so you can move them far enough apart, in theory, that we can now image them and tell them apart in microscopes. You can also see this in action with a real piece of brain tissue here. This is a piece of mouse brain tissue, and this is sped up, but I hope you can see it swelling. The whole process takes an hour, not a minute, so this is sped up by about 100-fold. But it's a piece of mouse brain tissue that will swell about 100-fold in volume, about 4.5 times in each direction with this expansion microscopy process. So what do these things look like? Well, on the left is a piece of, in this case, mouse brain tissue. From top to bottom is zooming in more and more. The green is labeling neurons with a glowing dye. And then the purplish blobs are actually neural connections. So the left-hand column is before expansion, and it's really hard to see. The right-hand column is after expansion, and you can see a lot more detail. In fact, if you zoom in into this little purplish blob, you can see that it's not a purple blob. There's a blue part and a magenta part. And those are the 2 sides of a synaptic connection in the brain. And the distance is exactly as you would expect it to be, except scaled up by the expansion factor. So one thing we're doing now is trying to use this to map brain circuits. How does information flow, and how does it get transformed as it flows from sensation, and it gets integrated with memory and emotion and leads to decisions and actions? And also, can we map brain pathology? Can we find out how things change? Now, one of the nice things about moving all these molecules apart is you can run interesting chemical reactions in there. And this is a genomics meeting. One of the things we're doing, actually in collaboration with many groups, is to get sequencing technologies to work in these expanded samples. And so working with George Church's group, which pioneered this in situ sequencing methodology, where you can actually sequence DNA and RNA right there in the cell, if we decrowd these molecules and move them apart, we can run these reactions with much higher yield and get a lot more information. So we can look at the genome perhaps, or right now we're focusing more on the RNA, the expressed genes, to figure out the recipes of how these cells are and what makes them different from each other. And furthermore, when you expand these things, you fill them with water, so they're completely clear, and so you can image them, as shown in the middle bottom here, with extremely high-speed automated systems. And so that's our hope, to really allow the full omic interrogation of tissues. Why can't we see where these different biomolecules are with nanoscale precision throughout cells, throughout tissues? Now, when you build something to tackle brain disorders, you often find that it's useful for many other things. And if you think about it, many of the other really difficult to tackle diseases in the body are related to large 3D objects that originate with nanoscale pathology and lead to disease. And so cancers, changes in the immune system, aging-related changes, and so forth. And so one thing we've been doing is developing strategies that might be useful for diagnosis and pathology. Why can't you take a biopsy, expand it 100-fold in volume using the technology that we developed for brain mapping, and you would just get a lot more information by doing that? So we collaborated with Andy Beck's group and Astrid Wine's group and other pathologists at Harvard Medical School and developed a pathology-optimized version of expansion microscopy. And then we could take classical, as shown in the middle row here, stained tissues and compare them to the expanded tissues and see whether we could do better automated pathology on the computer. So what we found in this little study, we worked with samples from 100 breast cancer patients and tried to classify the lesions. Some are benign and don't even require any intervention. Some are severe and require intervention and treatment. And so by expanding them, we found that we could actually train this machine learning algorithm to better classify the cancers or precancerous or benign lesions much better than if you had to operate with the normal resolution samples. So by blowing it up 100-fold in volume, you get a lot more information that allows you then to recognize the morphology and molecular composition and so forth of biological specimens. And we have data also showing that we can improve the ability of light microscopes to diagnose other kinds of diseases like nephrotic kidney diseases, for example. So to summarize this first part of the talk, we developed a way to isotropically and evenly expand biological specimens, and we're very hopeful that this will help us pinpoint causes of diseases and help us understand the brain, but also in the short term might also serve as a very practical way of improving diagnosis in the pathology context. And we're very excited about learning more about other disease areas and how we can possibly help. So in the second part of the talk, I want to talk about time. So of course, everything I showed you only works on preserved tissues, which is a euphemism for dead tissues. And if we want to deal with the high-speed operation of the brain, we have to also have dynamic ways of interrogating neural circuitry. And so one of the things that we've been working on is a way to control the high-speed activity of brain circuits. So again, these neurons are firing off high-speed pulses of electricity. If we could dial in information and enter it and activate cells and patterns, could you figure out how cells trigger disease states? Or maybe if you trigger the cells in the right pattern, could you heal a condition? And of course, if you can activate sets of cells, then you could also do interesting just basic science studies. What triggers the recall of a memory or the formation of a memory and so forth? So starting around the year 2000, Carl Deisseroth, then a fellow student, and I started brainstorming about whether you could use light to do this. And so the concept that we came up with, now known as optogenetics, was could you take, for example, solar panels and install them in neurons? Light shines in the solar panels, that converts the energy into electrical signals, and you can then control those electrical computations. Then you can put optical probes into the brain. The brain doesn't feel pain, and you can put, just as people put deep brain stimulators into human patients, and other kinds of in conditions like Parkinson's disease and other diseases, we could try to put optical probes in. The hard part, of course, is how do you get a solar panel into the brain cell? And so it turns out that all over the tree of life, you can find molecules that sense light and convert them into different electrical signals. So here's a single-celled green algae, and it has a little eye spot shown in brown in the middle, and it senses light and steers little flagellae to steer around in ponds so they can photosynthesize better. And if you zoom into that eye spot, you would find these molecules that, interestingly, sense light and convert them, the light energy, into signals that are electrical in nature. And so we're very lucky. It turns out that the molecule that does this is a protein. And that protein is encoded by a fairly short gene. And if we isolate the gene and put it into a neuron using any one of a number of strategies, then we can actually try to make that neuron sensitive to light. And so that's what Carl and I did starting around mid-2004 or so. We got the gene from a colleague who had played a role in originally discovering it, Geert Nagel. We put the gene in using transfection or viruses and so forth into neurons, and then hoped for the best. And serendipity struck. We actually found out that neurons could express this gene, and despite the fact that these genes come from single-celled critters and bodies of water, the neurons can manufacture the protein properly. Maybe even more remarkably, they could install them in the right spot on the outer membrane. And then maybe even more remarkably, they have the right speed and amplitude of effect so that when you shine light on these cells, they will respond with high speed, high fidelity electrical pulses. So now you can do things like try to put these molecules into certain cell types and see what you can affect. So for example, there's a cell type known as the basket cell, which is atrophied in patients with schizophrenia. It's a little star-shaped cell. If you express these genes, these opsins as they're known, in the basket cell, using any one of these methods of gene therapy, and maybe using some gene editing strategies in the future, then when you shine light on this brain circuit, even in the living brain, those cells will be driven by light, but their neighbors will not be light sensitive. So people are using this technology to try to discover all sorts of interesting pathways at the basic science level in the brain, but also trying to figure out, are there therapeutic activity patterns that you could drive that could affect interesting benefits in human health? I'll just give you a couple examples. Some people are trying to study the basis of addiction. Can you pinpoint the circuits in the brain that lead to that? And so in work that was led by Chris Fiorillo's group, they looked at neurons called dopamine neurons. And I think a lot of us have heard of them. They're often referred to as the pleasure center of the brain. But of course, as everything in neuroscience, it's much more complicated than that. So using a genetic therapy strategy, they equipped mice so that their dopamine neurons would be light sensitive, and then made a very simple behavior task. If mice went to one point in a box and poked their nose into a little sensor, they would get a pulse of light from a laser down a fiber aimed at these dopamine neurons. If the mice go to another point, a neutral point, then nothing would happen. And here you can see the mouse. It pokes its nose into a little sensor, gets a pulse of blue light, does it again, does it again. I think you're getting the picture here. So the mouse is basically working for light. And this is basically a way that you can prove that a specific site in a brain or cells at a certain site can causally drive a certain pattern of behavior, one in this case that might be maladaptive. So one of the things we've been doing is discovering more and more classes of molecule. We found molecules that we can put into neurons so you can turn them off with light. And many groups are exploring those for example, could use them to shut down epileptic seizures, for example, currently all still in animal models of neurological disease. But as an example, Akihiro Yamanaka's group was investigating narcolepsy and wanted to figure out, could you really prove that turning off a set of cells would cause an animal to fall asleep? And so they made transgenic mice that expressed these inhibitory opsins in a very small cluster of cells that make a protein called hypocretin or orexin. And these cells are atrophied or die off in patients with narcolepsy, and then they fall asleep at inopportune times. But could you really turn them off and figure out, is the causality direct, or do these cells die and then some other changes occur? And that causes this episodic sleepiness. So here's the data. You can see the percentage of the mice that are awake on the vertical axis and time on the horizontal axis. This molecule is sensitive to yellow light, not blue. And as you can see what happens, they turn on a laser, the light goes down a fiber aimed at these cells deep in the brain, and bam, all the mice are completely asleep in half a minute, and then you turn the light off and they wake back up. But you can see they're a bit groggy and they pass out again after another half minute or so. So you can tease apart the time dynamics. This is something that cannot be done just by looking at static loss of the brain. But maybe by looking at the time dynamics, one can learn more about the circuits involved. And even though these cells are lost in patients with narcolepsy, maybe you can find other circuits in the brain that you could restore function with. In our group, we were trying to push these molecules to their physical limits. For example, we recently showed that we could discover molecules from natural organisms that are sensitive to red or even near infrared light. So in the lower left, you can see that we could shine light that's 735 nanometers. That's 100 nanometers redder than a red laser pointer. And we could put an opsin into these cells and cause them to respond to these pulses of light by firing action potentials. And so one of my hopes here is that we're going to start to see more and more connections between these tools that give you very deep insights into complex 3D systems that lead to better diagnostics and therapies. And maybe I'll close in the last minute or so and just tell about one story that my group was recently involved with. We started working with Li Wei Sai's group at the Pickauer Institute at MIT. And her group was leading a study on trying to figure out if you could use patterns of activity to help alleviate Alzheimer's in Alzheimer's model mice. And so using the optogenetic tools, they discovered, with a bit of help from us, that if you pulse neurons, the basket cells that I showed you earlier actually, at a very specific frequency, 40 pulses a second, a series of biochemical changes occurred that caused the microglial cells of the brain to go and help clean up the amyloid plaques and some of the other hallmarks of Alzheimer's disease, in mice anyway. And of course, optogenetics requires a gene therapy. Is there a way that we can take these scientific insights and use them to inspire very practical and deployable therapies? And so what the team did next was to think about whether you could drive these 40 hertz or 40 cycles per second brain activity patterns through sensory stimuli. And so Li Wei's group pulsed an LED on the cage of mice while mice were just walking around. These are unmodified mice, no optogenetic tools in them. And if you pulse the LED at 40 hertz, 40 cycles a second, then that was enough to drive activity patterns in the brain that were in the same frequency range, and also to cause the brain to clean up its amyloid and other Alzheimer's hallmarks. So she and I just co-founded a company, Cognito, to try to see if we can do this in humans. Could you imagine, a visual stimulus, a movie even, where one would watch it, and that would trick the brain into cleaning up the plaques and other hallmarks of Alzheimer's disease? So I'll end there, and just want to acknowledge this is all a very, very large collaborative enterprise, and there's not enough time to go through all the people on this slide. But I want to point out that this is a very omnidisciplinary activity, neuroengineering. We have inventors and doctors and scientists and experts of every part of science, from physics to chemistry and beyond. And it's really that synergy that I think is equipping us to have the courage to go and tackle something as complicated as the brain. So thank you very much.